Welcome back BioSquad to another video. This is the 9700 October-November 2019 paper 1-3. If you are new, you got to join the squad right now by hitting that subscribe button. Before we continue, it would mean a lot if you could like this video, share and leave a comment down below. Let's then get into it. Question 1. Plant cells are stained and then seen with a simple light microscope using daylight as the only light source. Which cell structures are clearly visible at a magnification of 400? A. Chloroplast grana, B. Lysosomes, C. Nucleoli, D. Ribosomes. When stained and viewed with a simple light microscope, the only structure that would be visible clearly would be nucleoli. Therefore, the answer is C. Ribosomes are the smallest structures, hence cannot be seen clearly. Lysosomes are also quite small. Chloroplast grana is within the chloroplast and therefore cannot be visible through a light microscope. Hence, the answer is C. Question 2. How many nanometers are there in 1 millimeter? Well, 1 millimeter is equal to 1000 micrometers and 1 micrometer is equal to 1000 nanometers. Therefore, if you do the simple math, you will get 1 millimeter is equal to 1 followed by 6 zeros, that is 1 million nanometers. Hence, the answer is D. Question 3. Which cell structures contain 70S ribosomes? Structures that have 70S ribosomes are the chloroplasts, mitochondria, and the prokaryotic cells. Therefore, if you go to the answers 1, 2, 3 and 4, the obvious and correct answer would be 1 and 2, chloroplasts and mitochondria. Nucleus and cytoplasm? Well, nucleus doesn't contain ribosomes but rather makes ribosomes. Cytoplasm would have 80S ribosomes, not 70S. Hence, the answer is 1 and 2, that is A. Question 4. The electron micrograph shows part of a plant cell. Which combination correctly identifies labeled cell structures? I have labeled towards the sides what each structure represents. P represents the cell wall, Q represents the vacuole, R represents the chloroplast, S represents the nucleolus, as S is within the nucleus. Hence, if you go to the answers A, B, C and D, with S being the nucleolus, you would get the answer either being C or D. And yes, P is the cell wall, Q is not the cytoplasm but the vacuole. As you can see, it is quite different compared to the surroundings, hence should be a vacuole, as cytoplasm would contain many other organelles. Therefore, the answer is D. Question 5. 4 students were asked to match the function with the appearance of some cell structures in an animal cell. The functions were listed by a number. 1. mRNA passes through the ribosome. 2. Organizes microtubules to produce a spindle during cell division. 3. Packaging of hydrolytic enzymes that will remain in the cell. Function 1 occurs during transcription in the nucleus. Therefore, if you are matching it with the correct appearance, it should be a double membrane interspersed with pores, because a nucleus has a nuclear envelope, or rather 2 membranes, hence has a double membrane, and of course it has pores called the nuclear pores. Therefore, function 1 should match with X, leaving us with the answer being either C or D. If you look at function 2, organizes microtubules to produce a spindle during cell division. That is obviously done by the centrioles. And how do the centrioles appear? Well, the centrioles are non-membrane bound cylindrical structures. It is not non-membrane bound spherical structures, as non-membrane bound spherical structures represent the ribosomes, not the centrioles. Therefore, function 2 matches with the appearance Y, non-membrane bound cylindrical structures, leaving us with the answer being D. 1 matching with X, 2 matching with Y, 3 matching with Z. Question 6. Tests for biological molecules were carried out on food samples. Which correctly matches the biological molecules in the food samples with the color for a positive result? The keyword over here is positive result. The positive result for starch is blue-black, and the positive result for fructose is from a color scale from blue, green, yellow, orange and finally red, where blue means there is no reducing sugar present and red means there is reducing sugar present of the highest concentration. Therefore, if you go to the answers A, B, C and D with starch matching to black, you would get the answer between A and B. And considering between answers A and B, if you look at fructose, there is no color called white, hence answer A is incorrect, leaving us with the answer being B. Fructose matching to red, starch matching to black, and yes, triglyceride does match to white because triglyceride comes under fats and for fats it is a milky emulsion, which is the positive result. Therefore, the answer is B. Question 7. What occurs during the formation of a glycosidic bond between two alpha glucose molecules? A. A 1,4 bond is always formed. B. A hydrogen bond is always formed. C. A molecule of water is always formed. D. A hydroxyl group is always formed. Answer D has obviously nothing to do with the formation of a glycosidic bond. If you look at answer C, a molecule of water is always formed. That is correct because formation of bonds is related to condensation reactions, and in condensation you would get a water molecule being produced. Therefore, answer C is the correct choice. Let's look at answer B. A hydrogen bond is always formed. Well, that is also incorrect because in glycosidic bonds, hydrogen bonds are not involved. Let's look at A. A 1,4 bond is always formed. That is also incorrect because between two alpha glucose molecules, a 1,6 bond could also be formed. Therefore, A is incorrect, leaving us with the answer being C. Question 8. What is the maximum number of condensation reactions that occur when a triglyceride is synthesized? When a triglyceride is synthesized, there will be 3 ester bonds, and formation of 3 ester bonds means there will be 3 condensation reactions. And when condensation reactions occur, a water molecule is always formed, as discussed in the previous question. That means the maximum number of condensation reactions would be 3. Therefore, the answer being C. Question 9. 3 proteins that have a quaternary structure are listed. Type IX collagen is formed from 3 different polymers. The main form of hemoglobin consists 2 alpha globins and 2 beta globins. HIV protease consists of 2 identical polymers. Which row shows the correct number of genes needed to code for each protein? If you look at HIV protease, it consists of 2 identical polymers. Since they are identical, they will be requiring the same gene, and hence it will be 1 gene required only for HIV protease, as 1 gene could code for both the identical polymers. Therefore, leaving us with the answer being either B or C. Let's look at the main form of hemoglobin. Contains 2 alpha globins and 2 beta globins. Although the total will appear to be 4, there will be 2 types of globins only. Hence, 2 genes for each one. 1 gene for the alpha globin and 1 gene for the beta globin. Leaving us with the answer being C. And type IX collagen would obviously contain 3 genes. Why? Because there are 3 different polymers. Hence, each gene would code for each different polymer. Therefore, the answer is C. Question 10. The diagram shows the structure of an amino acid with some parts of the molecule shaded. Which diagram shows the shaded part of the molecule that is common to all amino acids? Key thing to remember is that all amino acids have 3 constant regions, which are a hydrogen atom, a carboxyl group or a COOH group, and a NH2 group. Therefore, if you go to the answers A, B, C and D, the best possible answer is B. Hydrogen is common, therefore should be shaded. COOH group is common in all amino acids, therefore it should also be shaded. And NH2 is also shaded, therefore B is the correct choice. Question 11. Some extracellular protease enzymes are synthesized in an inactive form called zymogen. Zymogen is converted to the active protease in the Golgi body. What is the purpose of producing zymogen before converting it to the extracellular protease? Since it is an extracellular enzyme, the purpose of it is to be active outside the cell and digest proteins outside the cell. In order to prevent digestion inside the cell, it needs to be inactive. Therefore, the purpose of converting it into an inactive form is to avoid unwanted digestion of proteins inside the cell, leaving us with the answer being A. Why are the other answers incorrect? Let's look at B. To avoid unwanted digestion of proteins outside the cell. That is incorrect because digestion of proteins outside the cell is its main purpose. Hence, it should be active outside the cell. However, the question is asking why it is converted to an inactive form before converting it to an active form. Let's look at C. To catalyze digestion of proteins in the cytoplasm. That is also incorrect as cytoplasm is within a cell and the purpose of the enzyme is to function outside the cell, not within the cell. And answer D also has RER, which is within a cell, therefore it is incorrect. Question 12. The Michaelis-Menten constant for an enzyme catalyzed reaction is referred to as Km, and the maximum velocity of such a reaction is known as Vmax. Which statement about Km and Vmax is correct? A key thing you should remember is that lower the Km, higher the affinity towards the substrate and vice versa. Again, lower the Km, higher the affinity, higher the Km, lower the affinity. Let's look at the answers A, B, C and D. Answer A. An enzyme with a high value of Km can reach its Vmax at a low substrate concentration. That is incorrect. because high value of Km means lower affinity. Lower the affinity means higher substrate concentration required to reach Vmax, not low substrate concentration. Therefore, A is incorrect. B. An enzyme with a high value of Km has a high affinity. That is again incorrect. Higher the value of Km, lower the affinity, not high affinity. Answer C. An enzyme with a low value of Km can reach its Vmax at a high substrate concentration. Again, this is incorrect. Lower the value of Km, higher the affinity. Higher the affinity means less substrate is required to reach Vmax, not high substrate. Leaving us with the only answer being D. An enzyme with a low value of Km has a high affinity to its substrate. That is correct, as lower the Km, higher the affinity. Question 13. The diagram represents part of the fluid mosaic model of a cell surface membrane. Which parts of the membrane are hydrophilic? Structures facing the outer watery regions are hydrophilic, as hydrophilic means water loving. Therefore, if we go to the answers, structure 1, 2 and 3 are hydrophilic as they point out towards the watery surroundings. Whereas structure 4, it is hydrophobic because it is protected from the watery surroundings. Leaving us with the answer being A. 1, 2 and 3 are hydrophilic. Question 14. Which statements about the proteins and glycoproteins in cell surface membranes are correct? 1. They can allow cells to bond together to form tissues. 2. They can recognize messenger molecules like hormones. 3. They can be antigens and allow cell-to-cell recognition. Proteins and glycoproteins can bind cells together to form tissues, as tissues are a collection of cells. Therefore, statement 1 is correct. They can allow cells to bond together to form tissues. They can also act as antigens or cell surface membrane receptors, binding to structures such as hormones. Therefore, statements 2 and 3 are also correct. They can recognize messenger molecules like hormones. 3. They can be antigens and allow cell-to-cell recognition is also correct, because proteins in cell surface membranes are normally involved in cell signaling and cell recognition. Therefore, the answer is A. 1, 2 and 3. Question 15. Which pair of factors is inversely proportional to the rate of diffusion? A. Concentration gradient and surface area over which diffusion occurs. A is incorrect because higher the surface area, higher the rate of diffusion. Hence, they are not inversely proportional. B. Distance over which diffusion occurs and size of diffusing molecule. That is correct, because as the size increases, the rate of diffusion decreases. Why? Because when the size increases, the surface area to volume ratio decreases. Hence, if the surface area to volume ratio decreases, that means the rate of diffusion will also decrease. If you look at distance over which diffusion occurs, well, shorter the diffusion distance, higher the rate of diffusion. Therefore, they also have an inversely proportional relationship. Hence, the answer is B. Let's look at C. Size of diffusing molecule, yes. Concentration gradient is incorrect, because steeper the concentration gradient, higher the rate of diffusion. They don't have an inversely proportional relationship. D. Surface area over which diffusion occurs. Again, that is incorrect, because higher the surface area, higher the rate of diffusion. Question 16. Raisins are dried fruit that contain high concentrations of sugar. Which row is correct when raisins are first put into water? Raisins, since they contain high concentrations of sugar, they will be more negative, as more negative means lower water potential. When there is high concentrations of sugar, there will be low water potential within the raisin. Hence, water potential in raisin compared to surrounding water should be more negative, leaving us with the answer being either C or D. Direction of water movement. Since the concentrations of sugar within the raisin is higher than the outer surrounding water, water will move into the raisin because a water potential gradient is set up. Since the raisin is more negative, water will move into the raisin from a region of higher water potential to a region of lower water potential, leaving us with the answer being C. Question 17. Which processes that occur in cell division are represented in the diagram? I have marked over here what occurs as the time passes. Since the mass of nuclear DNA in a cell increases, DNA replication will occur, therefore leaving us with the answer being either A or B. And cytokinesis does occur, because after nuclear division, which is marked over here, there will be a time where the cell divides into two daughter cells, and that time is shown by the mass of nuclear DNA in a cell being constant as the time proceeds. Therefore, that is cytokinesis. Hence, the answer is B. DNA replication is shown, nuclear division is shown as the mass of nuclear DNA decreases, and then cytokinesis is shown due to its constant mass as the time proceeds. Question 18. What happens to the telomeres during mitosis of a cancer cell? Cancer cells are able to undergo repeated cell division by mitosis, hence it would require the telomere length to stay the same, because if the telomere length changes, that means it will affect its repeated cell division ability. Therefore, the best answer is D. They stay the same length. Question 19. The graph shows 3 measurements obtained after metaphase of mitosis. What measurements do the curves represent? Keyword over here is after metaphase. After metaphase, the process is anaphase, where the centromeres split and the centromeres are dragged towards the opposite ends of the cell or the opposite poles of the cell. Initially, the distance between centromeres of sister chromatids would be 0, and after metaphase, it would increase as the centromeres are being pulled towards the poles of the cell. When they are pulled towards the poles of the cell, the distance between the centromeres of sister chromatids would increase, representing curve number 2. Hence, leaving us with the answer being either A or D. Also, the distance between centromeres and poles of spindle should decrease as the centromere is pulled towards the poles, which is represented by the curve number 1. Therefore, leaving us with the answer being A. Distance between centromeres and poles of spindle 1. Distance between centromeres of sister chromatids represented by 2. Distance between poles of spindle represented by 3. Question 20. The diagram represents a nucleotide containing guanine. Which statements about this nucleotide are correct? 1. The carbohydrate can be ribose or deoxyribose. 2. Base pairing occurs with 3 hydrogen bonds. 3. Guanine is purine. Key thing you should know is that guanine and adenine are both purines. Therefore, yes, guanine is a purine. Hence, statement 3 is correct. Base pairing of A and T occurs with 2 hydrogen bonds, whereas of C and G it occurs with 3 hydrogen bonds. Therefore, yes, statement 2 is also correct. Base pairing of guanine occurs with 3 hydrogen bonds. Statement 1. The carbohydrate can be ribose or deoxyribose. All the other nucleotides apart from uracil can be ribose or deoxyribose. Only uracil can be ribose, leaving us with the answer being A, 1, 2 and 3. Question 21. What is the maximum number of codon-anticodon interactions within one ribosome? Within the ribosome, there are 6 exposed bases allowing 2 codon-anticodon interactions. Therefore, the answer is A. The maximum number of codon-anticodon interactions within one ribosome is 2. Question 22. Michelson and Stahl investigated DNA in bacteria. They grew bacteria in a medium with only heavy nitrogen until all of the bacterial DNA was heavy. These bacteria were moved from a heavy nitrogen medium and cultured in a medium with only light nitrogen. A sample of bacteria was collected from the first generation and their DNA analyzed. Hybrid DNA contains heavy DNA and light DNA. Which row shows a percentage of light DNA strands and the percentage of hybrid DNA molecules in the first generation produced in the medium containing light nitrogen? This, although a long question, you should break it down into separate parts. The keyword over here is first generation. Hence, they are looking at the first generation only, and they are asking about the percentage of light DNA strands and the percentage of hybrid DNA molecules of the first generation. And there is a difference between strands and molecules, as one molecule consists of 2 strands. So initially, both the strands would be 15N or the heavy nitrogen because it is first cultured in a medium with heavy nitrogen. Once it is removed from heavy nitrogen and placed in a medium with light nitrogen, that is 14N, there would be DNA replication occurring, and the first generation would consist of 100% of hybrid DNA molecules. What do you mean by hybrid DNA? It contains a strand of heavy DNA and a strand of light DNA. That is, one strand belongs to N14 and one strand belongs to N15. Therefore, as you can see towards the right, I have drawn over here the initial stage and after the first generation, how the process looks. After the first generation, there would be 2 DNA molecules, and those 2 DNA molecules would be hybrid. Therefore, 100% of hybrid DNA molecules. Percentage of light DNA strands. Well, percentage of light DNA strands would be 50% because there are only 2 N14 strands out of the 4 strands. If you do the math, you would get 50% because 2 divided by 4 times 100 would give you the percentage, leaving us with the answer being C. Question 23. Which statements about tRNA structure are correct? 1. There is a binding site for the attachment of a specific amino acid and different binding site for the attachment to the ribosome so that translation can occur. 2. There is a ribose phosphate backbone with strong phosphodiester bonds in areas within the polynucleotide chain where base pairing occurs. 3. there is an anticodon that contains the same triplet of bases as the triplet of DNA bases that has been transcribed to produce the mRNA codon. Statement 1 is correct. There is a binding site for the attachment of a specific amino acid and a different binding site for the attachment to the ribosome. Because in a tRNA molecule there will be two different sites. One site is for the attachment of a specific amino acid and the other site contains three bases representing the anticodon that binds to the ribosome. Hence statement 1 is correct. If you look at statement 2, there is ribose phosphate backbone with strong phosphodiester bonds. Yes, that is also correct, because since it is an RNA molecule, it will have ribose phosphate backbone and a nucleotide will contain strong phosphodiester bonds, and a tRNA molecule has a clover leaf shape. Hence there will be some base pairing occurring within the polynucleotide chain. Therefore statement 2 is correct. If you look at statement 3, it won't contain the same triplet of bases as the triplet of DNA bases, because in a tRNA molecule the bases are A, U, C and G. However, in a DNA molecule the bases are A, T, C and G. Therefore, if there is U within the anticodon, it will not be the same as the bases of a DNA molecule. Hence statement 3 is incorrect, leaving us with the answer being B, 1 and 2 only. Question 24. Which row explains why xylem vessels have no cytoplasm? Over here you simply have to know the key points of why xylem vessels do not have cytoplasm. It is because to provide a large volume for water to be transported and to allow faster water transportation. If you look at row number 3, to increase the cellulose content of the vessel walls, that does not explain why the xylem vessels have no cytoplasm, because cytoplasm not being present leads to the fact that it provides more space for water to be transported or more volume. Hence, more the volume, faster the movement. Therefore, the answer is C. Question 25. What causes water to move from the root hair cells to the endodermis? For water to move from the root hair cells to the endodermis, first it would be diffusion through the cell walls, followed by osmosis within the cytoplasm, leaving us with the answer being only A. Let's look at why B, C and D are incorrect. B. Diffusion through the symplast, osmosis and root pressure through the apoplast. That is incorrect, because osmosis doesn't occur through the apoplast. Osmosis occurs only through cytoplasm or through the symplast pathways. Therefore, B is incorrect. C. Osmosis from cell vacuole to cell vacuole, active transport in the endodermis. Active transport has nothing to do with water movement from root hair cells to the endodermis. Therefore, C is incorrect. D. Osmosis through the intercellular spaces, diffusion in cell walls and cytoplasm. Intercellular spaces is again related to the apoplast pathway, and osmosis doesn't occur in the apoplast pathway. Osmosis only occurs in the symplast pathway or through movement within the cytoplasm. Hence, the only possible answer is A. Diffusion through cell walls, osmosis down a water potential gradient in the cytoplasm. Question 26. What is true about a Casparian strip? The Casparian strip is formed from suberin, not lignin. Lignin forms only in the xylem vessels, leaving us with the answer being either B or D. And the effect of a Casparian strip is that it blocks the apoplast pathway, leaving us with the only answer being B. This location is obviously in the cell wall of an endodermis cell. It is not present in the cytoplasm of endodermis cell, leaving us with the answer being B. Location, cell wall of endodermis cell formed from suberin. Effect is to close the apoplastic pathway. Question 27. Which xerophytic adaptations reduce the water potential gradient between leaf surface and atmosphere? 1. Roll leaves, 2. Hairy leaves, 3. Sunken stomata, 4. Fused stomata, 5. Fleshy leaves. Water potential gradient is affected by only 3 adaptations out of the given 5. Roll leaves, hairy leaves or leaves with trichomes, and the third adaptation is sunken stomata. All these 3 adaptations trap moisture within the leaf, thereby reducing the water potential gradient or the steepness of the water potential difference caused. Hence, the answer is B, 1, 2 and 3 only. Adaptation 4 and 5 doesn't have anything to do with the water potential gradient. Question 28. When sucrose is loaded into the phloem, it has to travel from mesophyll cells to a companion cell and then to the phloem. And many plants, proton pumps and co-transporter molecules are involved in this process. Which row shows the relative concentrations of sucrose in each type of cell? Since sucrose is loaded into the phloem, It enters by diffusion from companion cells. Therefore, companion cells should have the highest concentration of sucrose, leaving us with the answer being only B. Highest sucrose concentration is in companion cell, followed by phloem CF tube element, and least is in mesophyll cell itself. And why is that? Because sucrose is actively loaded into the companion cell from the mesophyll cells. Therefore, the relative concentration of sucrose within the mesophyll cells would be the least, leaving us with the answer being B. Question 29. The image shows 3 white blood cells labeled X, Y, and Z. Which row correctly identifies these cells? X represents kidney-shaped nucleus, Z represents lobe nucleus, Y represents circular nucleus. And if you go to the rows, monocytes have a kidney-shaped nucleus, neutrophils have a lobe nucleus, and lymphocytes have a circular nucleus, leaving us with the answer being C. Cell X, since it is a kidney-shaped nucleus, it belongs to a monocyte. Cell Y, since it is a circular nucleus, it belongs to a lymphocyte. Cell Z, since it is a lobe nucleus, it belongs to a neutrophil. Hence, the answer is C. Question 30. Which is the correct sequence for the opening of the valves in the mammalian heart during one cardiac cycle? If you recall the process of the cardiac cycle, it starts from the SAN or the sinoatrial node sending waves of excitation from the right wall of the atrium. Therefore, it should start with the atrioventricular valves. If you go to the answers A, B, C, and D, answers A, B, and C are incorrect. Why is that? Because when the SAN that is present on the right wall of the atrium sends waves of excitation through the heart, both the left and right atrioventricular valves open at the same time, and that is only shown by sequence D. All the other sequences shows the opening of the left and right atrioventricular valves at different timings. Hence, they all are incorrect, leaving us with the answer being D. Left atrioventricular valve and right atrioventricular valve, they open at the same time, followed by aortic semilunar valve and pulmonary semilunar valve opening at the same time. Question 31. Red blood cells may contain a molecule known as 2,3-BPG. When 2,3-BPG binds to hemoglobin, a higher partial pressure of oxygen is needed to bring about 50% saturation of hemoglobin with oxygen. Which statements about the effect of 2,3-BPG are correct? Statement 2 is obviously incorrect. The binding of 2,3-BPG to hemoglobin reduces the Bow effect. It is actually the reverse. It increases the Bow effect because it leads to a reduced saturation of hemoglobin. As when it binds to hemoglobin, a higher partial pressure of oxygen is needed to bring about 50% saturation. That means the Bow effect increases. Therefore, answers containing statement 2 is incorrect, leaving us with the answer being either B or D. Why is statement 4 incorrect? When 2,3-BPG is absent, oxyhemoglobin is less likely to unload oxygen. That is incorrect because molecules that do not have 2,3-BPG are able to unload oxygen due to presence of other factors such as increase in carbon dioxide concentrations in actively respiring muscles. Therefore, 2,3-BPG is not a main factor that leads to oxygen being unloaded from hemoglobin. Therefore, 4 is not a correct effect. Effects 1 and 3 are obviously correct. Let's look at 1. 2,3-BPG in red blood cells causes the oxygen dissociation curve to shift to the right. That is correct because when it binds to hemoglobin, since higher partial pressure of oxygen is needed to bring about 50% saturation, that means the Bow effect increases. When the Bow effect increases, that means the curve shifts to the right. And effect 3, the binding of 2,3-BPG lowers the affinity of hemoglobin for oxygen. That is correct. Why? Because when it binds to hemoglobin, a higher partial pressure of oxygen is needed to bring about the same saturation with oxygen. That is 50% saturation. A higher partial pressure of oxygen is needed. Hence, the affinity towards oxygen is lowered due to the effect of 2,3-BPG binding to hemoglobin. Therefore, the answer is B, 1 and 3. Question 32. Aortic stenosis is a heart valve disorder in which the aortic semilunar valve opening is narrow. Which effect could aortic stenosis have on heart structure and function? A. The cardiac muscle of the left ventricle wall is thinned by blood leaking out of the left ventricle during ventricular diastole. That is incorrect because the left ventricle wall is not thinned but rather the opposite. It thickens. Therefore, A is incorrect. B. There is less cardiac muscle in the left ventricle and reduced diastolic blood pressure caused by the smaller blood volume entering the left atrium. The heart valve disorder occurs only to the aortic semilunar valve, which becomes narrow. Therefore, it doesn't cause any change to the blood volume that enters the left atrium. Hence, cannot be a smaller blood volume entering the left atrium as the blood volume remains unchanged. Therefore, effect B is incorrect. C. The tendons of the heart valves are weakened by blood being forced back through the left atrioventricular valve into the left atrium. The disorder occurs only to the aortic semilunar valve, not to the atrioventricular valve. Hence, backflow is prevented and blood cannot enter into the left atrium. Hence, C is incorrect. D. The wall of the left ventricle thickens, leading to an enlarged heart and inability to relax and fill completely during diastole. That is correct, because aortic semilunar valve is present on the left side of the heart. Hence, if blood is unable to pass through the valve due to the valve opening being narrow, the left ventricle thickens in order to provide more blood pressure to force blood through the semilunar valve. Hence, D is the correct answer. Question 33. What explains why the red blood cell count of humans increases when they remain at high altitudes? Red blood cell count is mainly linked to the reduced partial pressures of oxygen at high altitudes. In order to compensate for the reduced partial pressure of oxygen, RBC count or the red blood cell count will increase. If you go to the answers A, B, C and D, the best possible answer is C. The percentage of oxygen in the air has decreased. In other words, the partial pressure of oxygen has decreased. Question 34. Which may contain ciliated epithelium? Ciliated epithelium is only present on trachea, bronchus and bronchioles. Why is it not present in alveoli? It is because alveoli have a different type of epithelium known as the squamous epithelium. Hence, answers having alveoli are incorrect, and the answer should contain all the three structures, that is trachea, bronchus and bronchioles, leaving us with the answer being C. Question 35. Which row shows the effects of chronic bronchitis? This question right here simply involves memorizing the correct points. There would be an infection present, of course, leaving us with the answer being either B or C, and the lymph glands would be swollen, not destroyed, because T cells would be produced. Hence, the answer is C. Lymph glands swollen, alveoli damaged, bronchi scarred, infection present. Question 36. The statements refer to the disease tuberculosis. 1. The pathogen lives inside human cells, so is not accessible to the immune system. 2. The bacterial pathogen reproduces slowly. 3. The pathogen is not very sensitive to antibiotics, which explains why antibiotic treatment for TB takes a long time. Statement 1 is not a correct statement. It is because TB mostly lives in macrophages. Hence, it is accessible to the immune system, as macrophages are part of the immune system. Therefore, statement 1 is incorrect, leaving us with the only possible answer, that is D, 2 and 3 only. Statement 2 does explain, because when the bacterial pathogen reproduces slowly, the antibiotic treatment will take a long time, because antibiotics affect the pathogen at different stages of their cycle. And if you look at statement 3, the pathogen is not very sensitive. Yes, because the pathogen that causes tuberculosis has a method of avoiding contact with the antibiotics, hence it does take a long time. Therefore, D is the correct choice. Question 37. Scientists study the multi-drug resistant bacterial infections in children caused by one type of bacteria between 2007 and 2015. The percentage of multi-drug resistant infections rose from 0.2% to 1.5%. What was the percentage increase in multi-drug resistant infections between 2007 and 2015? How do you calculate the percentage increase? Well, the percentage increase is found by the difference, that is 1.5% minus 0.2% upon the initial percentage in the year 2007, that is 0.2%. Therefore, the difference upon the initial value, that is 0.2% into 100, will give you the percentage increase, and that answer is 650%. Question 38. Why is it necessary for a person with a bacterial infection to take antibiotics at evenly spaced time intervals? Over here in this question, it is simply so to maintain the concentrations of antibiotic inside the body, which is lethal to the bacteria, and no other answer explains why a person should take antibiotics at evenly spaced time intervals. Therefore, the best answer is B, to maintain a concentration of antibiotic in the body which is lethal to the bacteria. Question 39. The enzyme telomerase prevents loss of telomeres after many mitotic cell cycles. 3 types of cells are used to produce monoclonal antibodies. 1. Cancer cells undergoing uncontrolled cell division. 2. Mature B lymphocytes. 3. Hybridoma cells. Which cells involved in this technique contain active telomerase enzyme? cells that undergo multiple cell divisions by mitosis requires active telomerase. And cell number 2, mature B lymphocytes, they are unable to undergo multiple cell divisions, whereas the others can. Therefore, answers having 2 in it are incorrect, leaving us with the answer being C, 1 and 3 only. Yes, hybridoma cells are able to undergo multiple cell divisions by mitosis, and yes, cancer cells, they do undergo uncontrolled cell division, as mentioned in the question. Therefore, C is the correct choice. Question 40. What explains why monoclonal antibodies are useful in diagnosis of disease? Monoclonal antibodies are used in targeted therapy due to its specificity towards a pathogen, and they do not bind to antibodies. Therefore, answers having binding towards antibodies are incorrect, leaving us with the answer being either B or D. And if you look at D, their shape is complementary to one specific antigen. Yes, that is correct, because monoclonal antibodies are useful due to its specificity in targeted therapy as their shape is complementary to a specific antigen. Hence, the answer is D. Answer B, they bind to and neutralize one type of pathogen, does not explain why they are useful in diagnosis of disease. Answer D is more specific. Therefore, answer D does explain their use in the diagnosis of disease. That is the end of this paper. Thank you if you made watching it this far. Let me know which papers I should be doing next, and let's keep the bio squad growing.